Very, very warm welcome. I'm very, very pleased to be joined by Om this morning. Hello, Om.
Hello.
So, could you tell us a little bit about where you're from, first of all, and what your role is at the university?
Yeah, I'm studying for a PhD in bioinformatics at the University of Grenoble, which is in France. It's a nice mountain town. You can do lots of skiing and stuff, and a bit of study, of course. And so, yeah, me and my team, we look at viruses. We analyze data from a hospital, the Grenoble University Hospital, and we look at viruses like HIV and hepatitis C.
Cool. So I think a lot of us have heard about viruses. I was wondering if you could tell us a little bit about viruses, what exactly they are.
Okay, so the viruses I'll be talking about, those are not the computer ones. They are a little bit easier to manage than these. So a virus, it's a microorganism which infects. So technically it's not living outside it's not alive outside a living body, but once it gets into a host, a living tissue, it can use the host's resources and replicate itself. So and it causes quite a few diseases, like you must have known of HIV, and probably now the worldwide epidemic, not worldwide, but in Africa you have Ebola. So
yeah, so that's an interesting concept, that it's something organic, but perhaps we don't even know if it's alive or not.
Yeah, technically they aren't alive outside a human a living tissue.
Oh yeah, interesting. So something that strikes me about that is when we have living organisms, a lot of us think about different species. Mm-hmm. Can you have different types of viruses, different species of virus?
Yeah, you technically can't use the definition of species for a virus, because the most common definition for species is a set of individuals which can reproduce and produce fertile offsprings. But viruses don't do that. What they do is replicate instead of reproduce. So you use a term called quasi-species for a set of viruses, because they're almost a species. They are not a species. So when a virus infects a living tissue, so within the host they form a population of viruses which are just copies of itself, and they're slightly different from each other. Mm-hmm. And that set of viruses, it's called a quasi-species. So each virus type, so if you have HIV, if you have Ebola, all of them, it's just one type, but they are the set of the viral population within the host. It's called a quasi-species.
A quasi-species. So that's something that I think we're going to be all taking away today. That's a new word for me, anyway. Right. Quasi-species. So what are you interested in finding out about them? What are you learning as you're studying these quasi-species?
Right. So the thing about this viral population is it has several. It's a really dynamic population. So if you have a treatment, so we have treatments for HIV which don't remove the virus, but they control the virus enough to make life easier for the patient. So you have this treatment, but we see that it's not effective in all of the patients. So if you have 10 patients with HIV, the same treatment won't work equally, won't be effective for all of them. So what we are trying to see is, can we get some information by studying this quasi-species and try to relate that a feature of the quasi-species and relate it to the outcome of the treatment. So you get in a new patient, you see their sequences, see the viral population, and then maybe you can try to say something like, maybe the treatment, which it will fail after 6 months, or it will be effective, and this will help us in improving diagnostic procedures and making things more cost effective.
Yeah, so in terms of especially what's happening at the moment with Ebola, etc., this sounds like really, really important work. It's very applied work, very applied research.
Yeah.
Cool. So how about when you are actually doing that research, what do you actually do? How do you study those quasi-species?
Okay, so what we do is we need to get the genetic sequence from of the viral population. So we use a machine, it's just called a sequencer, and the most common ones are the size of a printer, an office printer, and they're equally complex. So so what we do is we put in the biological sample. So if you want the sequence of humans or plants or viruses, you put that in the sequencer, and what you get as an output is the actual sequence. It's just a text file containing the sequences. Mm-hmm. So but the thing is, it's not so easy. You don't get a complete sequence at the end of the procedure. Mm-hmm. So I'll give an example using this book. So imagine this book is it's your complete sequence.
Okay.
It's the complete sequence you're looking at, say a virus, HIV. So this complete sequence, it's ordered by the index. If you want to look at a specific part of the book or a specific part of the sequence, you just go to the index and you just find out, okay, this is something I like. You get the page number, you look at it. But we can't do that from the data we get from the sequencer. Because the data you get from a sequencer, it's not indexed. So
Yeah.
imagine this book, it's over 1,000 pages, and if it was just 1,000 loose pages and they're just all scattered all over, completely randomly out of order. So you can't just go to the index and find what you want. So what we have to do is first arrange all these pages into a specific order, and then we can look at the information we are interested in.
So you're trying to almost create that index.
Exactly.
Yeah, wow. And that sounds like a big job and quite a complicated thing to do.
Yeah, that's where we use computers quite a lot, because it's a really time-consuming and a complex task. You can't do it manually. You have to use computational power to help your analysis, and that is pretty much what we do.
Fantastic. Well, that sounds like really incredibly important work, and really, really good luck with the rest of your studies. And yeah, I think it leaves me to say thank you very much for joining us, and we can give them a round of applause. Thank you.